you very much. A very interesting lecture to hear on the eve of my departure to India, where I will go in 3 weeks. There are several papers showing that the administration of aflatoxin to animals and the associated liver damage is free radical based. Both ochratoxin and aflatoxin toxicity at the level of the liver can be prevented with what? Melatonin. There are studies published showing that to be the case. It's never been tested in humans, of course, but on the basis of the animal studies, I guess when I'm in India, I'll be taking melatonin. Of course, hepatocellular carcinoma is the most common carcinoma in the world because of those. And cholangiocarcinoma of the bile duct is extremely deadly. And of course, that's also associated with aflatoxin. And hepatocarcinoma and cholangiocarcinoma are likewise reduced by melatonin. Again, there are publications of this, not in humans, of course, because they don't normally intentionally infect humans with these molecules, but it is potentially possible in humans as well. Well, I'm going to talk about phytomelatonin, helping plants to survive and to thrive. Yesterday I talked about their ability to upregulate melatonin in response to primarily abiotic stress, but they do in response to biotic stressors as well. And they're very effective in doing so. Again, I show you melatonin. I showed you melatonin yesterday. It hasn't changed in the 24 hours. I remind you that it's extremely old. It's ubiquitously present in every organism where it's been studied, and it has very strong antioxidant activities because of what I refer to as the antioxidative cascade, where melatonin plus its metabolites detoxify oxygen-related species that normally damage tissues. These are stromatolites. They are the best representation of the origin of cyanobacteria about 3.2 billion years ago in Shark's Bay in Western Australia. These are the remnants of cyanobacteria from eons ago. And I remind you that those very primitive, maybe nearly the first organisms on Earth, evolved melatonin. And they've passed it down to all subsequent organisms. The highest concentration of melatonin in any plant that has been measured to date is the pistachio nut. This is particularly interesting for several reasons. The pistachio plant is extremely hardy. It's a desert plant, live without water almost forever, tolerate cold temperatures to minus 10 degrees Celsius, or heat temperatures up to 50 degrees Celsius. The question is it hardy? And are plants generally hardy because of their ability to either upregulate melatonin or the fact that they contain high amounts of melatonin, which this species does? You'll notice in these 4 varietals of pistachio nuts, the concentrations are micrograms per gram. The closest to those are typically in the high nanogram range, and in some cases in the picogram range. But in the case of pistachio, and what this illustrates here, This is the graphical abstract from this paper. And what this illustrates, they are implying that the ingestion of these nuts, because of their high content, melatonin content, would have effects on the brain. Melatonin is very well known in models of Alzheimer's and models of Parkinson's disease to reduce the damage to the brain. In other words, melatonin is used in some cases as a treatment for these conditions because the loss of the dopaminergic neurons in Parkinson's disease is absolutely free radical based. Beta amyloid, the damaged protein, and of course also tau protein, hyperphosphorylated, also generates free radicals in terms of Alzheimer's disease. So this is not far-fetched. It's far-fetched in the sense that nobody's ever used these nuts to reduce it, and the implication is it may be healthy to eat plants that have a high melatonin content. I show this slide kind of as a joke. Everybody knows the crop circles in the UK. Well, a crop circle appeared on July 21st, 2011 in Wiltshire. This is the melatonin molecule. Believe it or not, this is the melatonin molecule. Some very clever crop circle makers with some degree of intelligence or knowledge at least about melatonin. To my knowledge, this is the only chemical structure that's ever been put in the form of a crop circle. I remind you, as I showed you yesterday, the endosymbiotic theory of the origin of mitochondria and chloroplasts are from bacteria. Photosynthetic and non-photosynthetic bacteria that were engulfed by ancestral eukaryotic cells that eventually evolved into the chloroplasts and the mitochondria, and these organisms that were involved have the enzymatic machinery to produce melatonin. We are very interested in the possibility that in every cell in your body that contains mitochondria, and that better be all of them, melatonin may be produced within mitochondria in particular. And if that is true, that would be remarkably important. That is the site of most free radical generation. Likewise, in terms of the chloroplast during photosynthesis, a lot of free radicals are generated. So those two organs, if they produce melatonin, and we are working very hard on that possibility, that would be extremely important. We've already known, for example, this is actually the work of Byung-Wang Bak in Korea in collaboration with us. We have isolated, in fact, one of the enzymes involved in the synthesis of melatonin. This is tryptophan. This is its conversion to tryptamine in a plant. Serotonin is shunted to the chloroplast, and the chloroplast has the enzyme that will convert, in fact, serotonin to N-acetylserotonin. So part of the enzymatic machinery for melatonin synthesis in a plant is in the chloroplast. Whether this is due to inheritance from its precursor bacteria, we do not know. But we're very interested in maybe other steps and other plants having this capability as well. I'm going to talk a little bit today about the resemblance of melatonin to indole-3 acetic acid, auxin. They are both from tryptophan, structurally very similar. And when melatonin was identified in plants, one of the first suppositions was, not by us because we were not plant physiologists, one of the first suppositions was that maybe melatonin has auxin-like effects. In other words, maybe melatonin will also promote growth. Well, there's now a great interest in this possibility by us and others, and we now know that. I'm going to talk about these. Melatonin improves seed germination. I remind you that seeds have an inordinately high concentration of melatonin to begin with, because it is the next generation, they have to be protected. It enhances growth and maturation of plants. I will show it improves product yield in the 3 species where it's been studied. Flower development and the rate of fruit development is impacted by melatonin. Here's an example of a melatonin deficient plant. In this case, Osaki and Azura in Japan genetically engineered a plant so it was enriched with a tomato plant that was enriched in indolamine 2,3 dioxygenase. This enzyme metabolizes melatonin very rapidly. So these plants with enriched 2,3 dioxygenase are relatively melatonin deficient. If the tomato plant does not have sufficient melatonin, are there any phenotypical changes in the plant? Indeed, there are. This is the normal tomato leaf. Terminal leaflet with 2 bi-leaflets in succession. After, I've got a solution. Ooh. It's hard to press one without the other. At any rate, as you can see, those that are deficient in melatonin, their leaves form very abnormally. There's a lot of different phenotypes, but none of them are very reminiscent of this. This looks relatively similar, except these leaves are flattened and less serrated. Conversely, a lot of them develop either leaflets in lieu of regular leaves, or they develop multiple leaves, or sometimes the second bipetal leaf is absent. Clearly, the loss of melatonin in these plants has an impact on their phenotypic appearance. This is a study we did in conjunction with a group in Beijing. I remind you that soybeans are maybe the largest commodity product in the world. 300 million metric tons of soybeans are produced every year. If there is anything you can do to enhance soybean production, short of planting them in every field throughout the world, it would obviously be an advantage. In this case, we added melatonin to the reagent, what is called a seed coating reagent. Soybeans are always placed in a seed coating reagent before they are planted. We added melatonin either in concentrations of 50 or 100 micromolar, planted them in pre-soaked soil in a greenhouse. I remind you that these are all under very controlled conditions, and looked at the germination. Normally, seeds germinate at about 60-65%. If they were infused with, I don't know if it was infused, it was applied to the outside, melatonin, then the seed germination increased to about 80%. This alone would be an advantage by virtue of the fact you'd waste a lot less seed soybean. And then if you look at the unifoliate leaf area as a consequence of these early plants, it is in fact larger. The plants seem to grow better. And of course, we measured a lot of other things like chlorophyll degrading enzymes and everything. And if you look at the plants themselves, the number of trifoliate leaves are greater and the plants are taller. Therefore, they develop more trifoliate leaves, and the terminal, the top third trifoliate leaf, which is commonly used as a measurement, is also increased in size. So the implication is that the seeds benefit from the application of melatonin. After 12 weeks, that was after 5 weeks. After 12 weeks, when the plants were mature, we, of course, looked at the plants. And again, there's obvious difference between the no melatonin and the 50 and 100 micromolar exposed plants from seeds. We then looked at the pods, the number of seeds per pod, and we measured the total weight of 100 seeds. Well, this is the number of pods on these plants. This is the average number of pods on these plants, and this is the average number of pods, and that data is represented here. The number of pods increases as a consequence of melatonin exposure. The number of seeds per pod likewise increases, albeit the seed size, if you measure 100 seeds, there's no difference in the size of the individual seeds, but there's more pods and there's more seeds. So in fact, the yield was greater because of plants grown that were treated with melatonin. As I say, there were 370 million tons. This enlargement, even if the enlargement is only a fraction of that, say 5% rather than 15%, the seed production in terms of total output would be very significant in terms of gross tonnage. A very small increase when you're harvesting 370 million tons, a 5% increase or a 2% increase is very significant. The other thing that is interesting, if you read the literature on soybeans, they anticipate no more soil will be dedicated to planting of soybeans. There's a lot of other crops that are competing. So we have to improve crop production on the fields that we have. The question is melatonin a possibility? Melatonin is very inexpensive, very easy to produce, in absolutely pure form. It's easy to administer, it has a long half-life, shelf life, and it has essentially no toxicity. We have currently a study going on in northern China, an outdoor study on a couple of hectares to determine whether, as I said, the other studies were done under controlled greenhouse conditions, optimal. Will this be practical in an open field situation? That's the question we are testing this summer. Margot Pozmik from Łódź, Poland, Margot. She also has looked at this in terms of several plant species. She does it a little bit differently. You cannot osmoprime, what she calls osmoprime, the soybean seeds. They have a different coat. But what she does is she osmoprimes seed corn or cucumber or bean overnight in a solution of melatonin, different concentrations, 15 micromolar up to 500 micromolar, and then she, after the following day, she measures the amount of melatonin in those seeds, and it's very rapidly taken up. So they are enriched with melatonin. She then plants those seeds outdoors. And she observes that mung bean response is very significantly better in terms of product with 50 micromolar melatonin incubation. Melatonin may, in fact, become toxic, or if you want to call it toxicity, if they're still normal, but it may become toxic at very high concentration. We've also seen that this concentration of melatonin for plants that may be toxic, but interestingly, the roots on these plants are better developed. In other words, melatonin doesn't inhibit root development, particularly lateral root development, but it does impede product production, which is kind of ironic. One would think with a better root system, there'd be a close parallel. But in fact, she finds, and this was a very small patch outdoors It wasn't a large field study, so the data must be considered preliminary, but are consistent with what we know from the soybean study that we did. And also there's one study done in Spain, in Murcia, Spain, by Arnau, who used not the mung bean, the lupin bean, and he found the same thing. In other words, infusing osmopriming seeds with melatonin allows higher germination, and Posmick showed that at all. I didn't show the slides. Higher germination, better growth, and increased product yield. This idea of lateral root primordia, or lateral roots on roots, are very important. They're very dynamic roots. They're very important for the health of the species. And this is a case where we looked at, or Tsong et al., looked at primordia, this is Feulgen-stained roots, and shown that primordia in, I think the concentrations, well, here we're 10 micromolar and 500 micromolar were increased. Remember I said the concentration of melatonin does not seem to impede lateral root growth. Posmick showed the very same thing. She showed the roots of these plants, and they were more robust in the 500 micromolar treated plants, but the product yield was less, which was, like I say, somewhat expected. But if you look at root primordia, in this particular case, you can see there are more root primordia identified here than in here, and likewise here than in here. So melatonin seems to promote lateral root and also adventitious root. And all genes associated with this change are in fact upregulated by melatonin. A lot of genes were measured in these animals. That's also seen here. This is a study, I should have acknowledged the authors. I don't have it on here. It's by Bak in Korea, a good colleague of ours. Here's melatonin synthesis. And these plants were transgenic for this enzyme, acetyltransferase, which converts serotonin to N-acetylserotonin, which then goes to melatonin. So they had more rapid melatonin. So here's an example of a plant that was producing a lot of its own melatonin in excess of normal. These seeds were then planted in 12 degrees Celsius for 8 days, cold stress, and the plants were examined. Well, you can see that the melatonin content in these plants was about 5-fold higher than the controls. Not surprisingly, we incubated them in melatonin. But likewise, there was a 2 to 2.5-fold increase in chlorophyll. You can actually see the difference between these two, and clearly the root development is likewise better. Now, we didn't follow these plants to maturity. These were only these young plants. But presumably, this would follow through over time, hopefully. Quality. Quality of fruit products is very important. In this case, this individual, Sun and Ren, Dr. Ren is actually in the United States at Virginia Tech. Somebody here was at Virginia Tech, I forgot. Somebody said they were at Virginia Tech. Do you know Dr. Ren? Ren? Maybe he wasn't there when you were. But at any rate, this study is done at Virginia Tech and in China. I haven't met him. I was hoping he'd be here, as a matter of fact, so I'm showing his work for him. But at any rate, they harvested green cherry tomatoes, kept them for 25 degrees at 15 degrees Celsius and 80% humidity. But when they picked them, they incubated for 2 hours, some of them in 50 micromolar melatonin or 100 micromolar melatonin. After 25 days, they measured lycopene, anthocyanins. They photographed them, obviously. And clearly, melatonin improves post-harvest maturation of these plants. In other words, here is, in fact, the lipokine content. You can see it increases substantially as a consequence of incubation in melatonin. We tried this with peaches, but we only dipped them in the melatonin solution for a very short interval. In this case, it was for a 2-hour interval. I don't know I suspect that's very critical for the melatonin to get somewhat into the plant. And if you can see also the other gene expression related to lycopene synthesis, synthase 1 and keratin isomerase, these are related to lycopene synthesis, and they are upregulated in the melatonin-treated plants as well. Of course, everybody knows that after harvest, fruit can ripen, but in fact, they can ripen faster in these conditions. And I don't know how to judge the quality of fruit. I know what I like. But they claim that the quality of this fruit was also enhanced, either measuring ethylene. I don't know what qualifies as quality. At any rate, here's another slide from the same group where they looked at anthocyanin accumulation after storage again, and anthocyanins clearly develop at a much rapid rate in those plants or in those products that are treated with melatonin. Anthocyanin accumulation, altered sugar metabolism, tissue softening. I presume that's a part of what quality is. And obviously, a half-life of product on the shelf is very, very important. I don't know what melatonin will do to half-life. It enhances, obviously, development, but it also prevents post-harvest physiological deterioration in cassava. Cassava is a very hardy root. It's a very important product in sub-Saharan Africa, for example, because it grows under the worst possible conditions. It's like the pistachio. It doesn't need much nourishment from the soil, and it will, but once you harvest it, typically what they've done in the past, because it deteriorates so rapidly, because of free radical mechanisms, you understand, they protect them from oxygen. They protect these roots from oxygen by putting in plastic bags so they're not exposed to a lot of oxygen, or they coat them with wax so the oxygen cannot get to the fruit. But if you cut this fruit, within a very short time it shows marked deterioration. It's like cutting an apple and it turns brown. Free radical damage, oxidation, exposure to oxygen. And if you add melatonin, you can see that that is delayed. So post-harvest physiological deterioration is delayed. Nothing remarkable at that because we know it's a free radical mediated process. So you would anticipate, in fact, that that's exactly what melatonin do. They were immersed for 2 hours in the melatonin solution at that concentration. Here's a proteomic analysis of a lot of this work. Again, Sun's name is down here. This is unfair. It should be up here where you can see it. But at any rate, a lot of things that melatonin does are summarized on this slide. We know melatonin is very protective against deterioration of plants by reducing scavenging directly reactive oxygen species, stimulating antioxidative genes. We can measure peroxidase, catalase, and all these other antioxidative genes in plants, and they are all upregulated by melatonin. I showed you yesterday a delay senescence, where you pick the leaves, incubate in melatonin, put them in the dark, and they deteriorate much. They refer to that as senescence. Different than I would classify senescence in an animal, but for plants that's called senescence. So delays senescence and prevents reactive oxygen species damage. It also upregulates enzymes, genes associated with signaling ethylene. Melatonin hastens ripening. Melatonin upregulates anthocyanin-related genes, and the product, this product at least, is improved as a result of melatonin use. I remind you that melatonin, very available. I have 5 pounds of pure melatonin because companies send me kilograms of melatonin. If you want melatonin, you know where to come. Does this have any practical applications? So there's melatonin in some plants. Big deal. We were asked to do a study with walnuts. Walnuts have a reasonably, I told you, that's a seed, of course. The seeds have higher concentrations of melatonin than do other parts of the plant. So we were commissioned to do a study using feeding walnuts or supplementing the diet of animals with walnuts to see what effect it would have in reference to something, and I'll say what that something is. But the nutrient value of the diets were absolutely rigidly controlled. Everything was equal except that one. Some of the animals were eating some walnuts. We ground up the walnuts and mixed them in the diet. So they could not, the animals could not say, oh, there's a walnut, I'm going to eat that. It was all mixed together. They didn't know when they were eating walnuts or when they weren't. But if you look at the serum melatonin levels, during the day they're normally low, 20 picograms per mil, very low in rats as in humans. We're kind of rat-like. And then after eating, of course, walnuts in the diet, the melatonin content increases, and it correlates with the total antioxidant status. In other words, the ability of this fluid to detoxify free radicals. That's what melatonin does very efficiently. We then injected, we gave these walnut diets to immune-compromised mice. They're nude. They have no fur. There's this rat strain that's immune-compromised called the buffalo rat, and they're frequently used because these are human cancer cells, LNCAP, human prostate cancer cells that are injected under the skin of these animals. They would normally be rejected, except these animals are immune-compromised. So the tumors grow. The tumors grow underneath the skin, and since they are nude, you can see this bulge appearing, and you can measure that tumor in terms of how large it is, and watch it grow. Well, what happened? After we inoculated the LNCAP human prostate, the same thing has been done with human breast cancer cells many times. Inoculated them here, within 3 weeks, one tumor became overtly apparent. In other words, when you inject the cells, you inject a couple million cells, but you don't see the bulge. But within 3 weeks, oh yeah, I can see that. There's a little bulge, and you can see it growing. And that was in the walnut animal. And after 7 weeks, another animal developed an obvious tumor, and then after 11 weeks, another did. But look what happened in the non-walnut treated animals. Many, many more. The tumors developed. So that in the final analysis, 44% of the tumors in those that did not have melatonin, so did not have walnuts, compared to only 19% in those that were consuming the walnut diet. If you look at the size of the tumors, that is also reflected there. These are the animals. There were only 3 tumors here, but they didn't grow very much. But those in the non-walnut treated animals, and you can see these tumors get very large. Here's one of the largest ones. That's this one right here. And that's the average size of these tumors. And some of them in the walnut-treated animals were also small. I mean, in the non-walnut treated. But generally, they were much larger, as represented by this line. So something in the walnut was stymieing the growth of these human prostate cancer cells. And I remind you that many similar studies have been done with female human breast cancer cells. We also looked at the livers to look at the total oxidative damage to the liver. Would melatonin reduce the oxidative damage? Presumably to all organs there was more melatonin, but we happened to take the liver and we measured F2 isoprostanes. F2 isoprostanes are not a normal measure. It's a very sensitive measure of lipid peroxidation, but it's expensive to do GCMS. So we did GCMS and F2 isoprostanes, and in those that were consuming the walnuts, the amount of liver lipid damage was very significantly reduced compared to animals on control diet, whether they had a tumor or not. So melatonin was functioning as, something was functioning as an antioxidant from these walnuts, but they have a significant high level. What we are doing now is we're repeating the study, giving a melatonin receptor blocker because we know the action of melatonin on prostate cancer is receptor mediated. Will these effects be blocked in terms of cancer growth by giving Luzindol, which blocks the main membrane receptors MT1 and MT2? Time. Okay. This is it. This is it. Okay. Here's what melatonin does. A lot of good stuff. Believe it. So, melatonin conserved in everybody, even you. Superior antioxidant. Vegetative actions. Maybe a prodrug in some cases. Remember yesterday I showed you it can be metabolized at 2-hydroxymelatonin. We don't know yet what that molecule does. Chloroplast and may synthesize includes proc yield in those that have been tested under primarily controlled conditions and has critical functions. The melatonin rhythm has been identified in the Moroccan guy. We know what the melatonin rhythm is. There's one lady here from Morocco I thought I'd point out. But at any rate, everybody. And it's always good. Thank you very much.
Any question?
Yes?
We got our first question for Dr. Reiter here.
Okay, doctor, my question maybe is not so scientific, but after your review, my question is, why do you think the agricultural industry is not really used these advantages of agricultural? Is not really used these advantages of melatonin?
Even the clinicians who have melatonin available do not use it as well as they should. Why? Who educates practicing physicians? The pharmaceutical industry. Yeah. They have drugs that will inhibit cancer. They're very expensive. They're not going to recommend melatonin, which cannot be patented, and is very cheap. It's a major, major problem. My cardiologist, my dermatologist, my internist, all recommend melatonin because I badger them so unmercifully, and I show them the literature. And I speak to continuing medical education people every year at a ranch in Texas, and they are also committed to melatonin. But in general, it's not exploited at all. That's why I'm invited to India, Sun Pharma, one of the largest companies in India, where I'm going in spite of the mycotoxins. I told you, aflatoxin toxicity at the liver, inhibited by melatonin. Good question.
Okay,
good question.
That's the same in agriculture with pesticide industry and fertilizer.
Yeah. I understand the problem.
Yeah, I agree with you, and in the sense of there is not more land to growth, and most of land is we are not available water also. Then it's important to understand this. And my second question is. Could be that melatonin is integrated as a base of a very, very big quantity of metabolites in plants and used for the plant to take advantage of the environment and to resist the different stress conditions.
You're absolutely right. I attribute it to melatonin. It's almost certain that all of the benefits of melatonin are due to secondary molecules that are generated by melatonin. And we know there are a lot of metabolites. I mentioned them yesterday, cyclic 3-AF, MK, 2-hydroxymelatonin. Furthermore, we know that melatonin acts synergistically papers are published showing it acts synergistically with vitamin C and vitamin E to enhance collectively the antioxidant potential. So in combination with other antioxidants, it's hard to say who's enhancing who. Is vitamin C enhancing melatonin, melatonin enhancing vitamin C? But together they're better. The product is greater than the sum of its parts. Let me put it that way.
I have some questions. Some questions. As you mentioned that 50 micromolar tomato fruit ripens. So what about the ethylene? It's a hormone, and ethylene is a hormone that ethylene. That yeah, it's a hormone. It is also responsible to ripen and to improve the quality of the tomato. And second question, it also, you mentioned that melatonin also works as a ABA, like a hormone in seed germination and plant growth. And third question, you also mentioned that in walnuts, it is present in large quantity and it can prevent from the tumors, cancer disease. So how you can differentiate, because once you are suggesting that it works like as a hormone and other in walnut, what about the fatty acids? There are some certain fatty acids are present in the walnuts. So how will you differentiate?
This definition of hormone is very, very difficult. I'll tell you a classic hormone by definition, but nobody calls it a hormone. Vitamin D3, produced in the skin, released in the blood, goes somewhere else and acts on receptors. That is a classic definition of the hormone. The problem with these definitions are very rigid. Melatonin has receptors which conventionally would make it a hormone. It has membrane receptors, MT1 and MT2. It has nuclear binding sites, the orphan nuclear receptors, and it binds the calmodulin and quinone reductase in the cytosol. So it has hormonal properties, but it's also a very efficient antioxidant which requires no receptor. If I'm a melatonin molecule and he's a free radical, I mutilate him immediately. So these terms are difficult. Now, I did equate the walnut study with melatonin, but that's a little bit unfair. We didn't prove that it was a walnut melatonin that caused the inhibition too. That's why we're doing the receptor study, blocking the receptors. There is ellagic acid also in walnuts in very, 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 very low concentrations. Ellagic acid is a known anti-tumor agent. There's a little bit of vitamin E in walnuts, which may have some there may be substances in walnuts we don't even know. So, these things are not as clear-cut. I present a great picture. But how much of it will prove to be valid under field conditions and so forth and so on. We are excited by it, and we are hopeful. Melatonin doesn't do any harm, fortunately.
Sir, I have a question.